So sometimes what you do, or sometimes if you just flip the bottom, yeah, that's like 1 plus. Yeah. So if you're not sure, just borrow for a second. So if you have a quick look up here, if you're not sure, remember the growth can settle down to the bottom over the week. So what you can do is hold it by the glass, never hold it by the lid, and just give it a quick flip with your finger, gently, and you'll see if there's any growth coming up.
This is pH study. Oh, no, this is pH study.
So there was?
Oh, did we have 2 pH 7? Is that different?
Yeah.
Oh, this one is still
That's like, that's
yeah, different bacteria.
This is
pH 5. This one is excuse me. So, this is Steph already. These are Steph. It's the same. I can compare. And Kim, I think we don't have your samples from over there, so because there were several labeled KH, there must be someone else with your. Oh yeah. Yeah, I think it does. This is clear. Yeah, this one's just clear. This one, I think it looks like it has a little.
I'm not good with it.
But not
Yeah. I think it's just not my name. Is it? And something else. Oh yes.
That's yours.
Temperature. All right. Temperature.
Okay, so let's find. Does anyone have a tube that is not theirs with E. coli that has the initials EW on it?
Yeah.
Double check your tubes originally if you had
No,
E.
they're all
coli.
different. These are all different.
Yours were missing?
Yeah, it's all different.
Oh, okay, or HH.
Oh, this one. Yes. So this is clear. This 2 maybe 2 plus. This is 1 3 plus. What is it? What is this on the bottom?
I think it's the bacteria. It's hard to tell.
Yeah, that's salt.
Something
It's just salt, yeah.
really.
It's just salt.
Oh,
This looks like yours. HH?
that's not.
Oh.
Katie.
We have two people with the same initials. I just connected the dots.
Really?
You and her have the same initials. Hazel Ann and Heidi. Yeah, they're left. So now that solves the mystery of where your tubes might be. You look like you have extra tubes. So if Hazel Ann, if you want to come over and figure out whose handwriting is who.
Yes.
It happens so rarely. It did not click in my head until that.
Next time you should try to
So if we use initials, maybe you want to use like H, like HHH, yeah, or HAH, Hazel Ann. Yeah.
Now what do we do again?
No, we looked at our own because those were labeled as a different.
We figured it out. The initials are the same. It's hard keeping this experiment organized too. So as soon as you have your data, don't stress about it too much. These are very imprecise calculations.
I'm trying to see like. But it's so weird that something is.
What do we
Doesn't
look
it look like something over there? No. I think it's just the way you swipe it. Yeah.
Okay, we're waiting for H. salinarium salt data. Staph aureus salt data.
We got.
Yeah, 5 degrees and 4 degrees are the same thing. It's just sometimes it's like, yeah, that's 4. Sorry. Refrigerators are about 4 to 5 degrees. Okay, and we're just waiting. Who has a step target for the salt? Okay. Pop up the data there, and at Cerevisier, we're waiting for 5% and 25%. And E. coli, we're waiting for 10%.
Hello, bacteria.
No. Yeah. That's like 2 pluses. Okay, so let's talk about this. Looks familiar, right? From the quiz? Okay, so E. coli. How would you classify E. coli by temperature according to this data? It's a mesophile, yeah. The middle range of temperatures, particularly body temperature and room temperature, is where you see growth for E. coli, so we call that a mesophile. What about Bacillus styrothermophilus? Thermophile, I love it when it's right in the name. And it worked. even at 55, sometimes it's too cold. It loves heat. It's a thermophile. Staph aureus? It's another mesophile, right? Optimal growth, you see the most pluses at 37 degrees. And B. subtilis? Also a mesophile. We work with a lot of mesophiles in this lab, which should make sense being that I'm usually telling you to incubate things in your drawers room temperature. Sometimes we want to give it a little warmer at 37, but very rarely do we incubate anything in the fridge or at 55. But given what you know about E. coli and Staph aureus in particular, do these designations make sense? Why? Correct, yeah. E. coli is usually found where in our bodies? What? The colon, yeah. E. coli, yeah. The intestines, the colon. Absolutely. What about Staph aureus? Where do we usually find Staph aureus? Sometimes in the throat, but more commonly, sometimes, but on our skin. Common skin bacteria. About 80% of all adults are colonized, meaning they have that bacteria present in their skin, or their nasal passages is the other very common place it lives. So it makes sense, it's a mesophile. Mesophiles are going to be mostly the ones responsible for things like human disease as well, because we are their optimal growth temperature. Close the door here. Okay.
Professor, what
Yes.
is that we use
Xerothermophilus, yeah.
Where does it live?
It lives typically in hot environments like hot springs.
Like, so it's like only.
It doesn't grow without 55 degrees Celsius, which is way hotter than our body temperature.
Yeah, but I think it's that.
Hot springs. So like, you know, you go to Yellowstone National Park, they have those geysers of hot water that pop up, or you go to like some of those thermal spas with like, you know, natural springs and you get to swim in them, popular in Europe, and you know there are some in Arizona and places like that, hot springs. We don't typically see them around our bodies or even campus. So yeah. Okay, so looking at things by pH, what is the vocabulary we're going to use to classify these bacteria? Acidophile, yeah. Neutrophile.
Alkali.
Alkalinophile, okay. So, E. coli is a neutrophile. What about Staph aureus?
Neutral.
Also a neutrophile, because the pluses are at 7. Usually though, I will say this data, a little different than what I expect. Usually Staph aureus likes things a little bit acidic, so I was expecting to see maybe 2 pluses here instead of 1. But these are qualitative measurements. So again, one person's plus is another person's plus plus, and they can really vary. The idea is that you all looked at a single organism, so you compile the data for that.
But usually
Yeah, I mean, no, usually it is a neutrophile. I was just expecting to see more growth at pH 5 and maybe no growth at pH 9. But it is still a neutrophile. If you think about it, Staph aureus lives on our skin. Your skin is slightly acidic. S. cerevisiae. Uh-oh. We can't classify that based on that data. But I will tell you it can go like that, so how would we classify it?
Acid.
Acidophile, right. And what about this last one? Neutrophile, right. Okay. Well, that's one way we can classify bacteria based on temperature, pH, and now salt. Let's have a look at it. I'm going to add theoretical data for one of these because there's no data here. So just know this is theoretical data. Okay, so E. coli. How do I classify It grew at 0% and
Huh.
and any other salt concentration.
Weird.
So what that tells me is the 0% column tells me if it needs salt to grow. Every other column tells me if it minds the presence of salt or not. So something that's halotolerant will grow at 0% and any other salt concentration. So I just want to check my microbiology here. Interesting. So typically, the E. coli data should look like this. How would I classify E. coli that way? Not halotolerant. Nothing wrong with how you analyze that, whoever did that organism, just giving you the other example. But typically, E. coli is not considered halotolerant. As cerevisiae, how do we classify that? Halotolerant, right? And what about H. salinarium? Yeah, it's an extreme halophile. It only grows at 25% salt. And this is actually an archaea. And if you remember from week 1, archaea are the extremophiles. They thrive in extreme environmental conditions. So H. salinarium is right in the name. Halobacteria, bacterium salinarium. So it has halo for salt and salinarium for salinity. So twice in its name. It loves salt so much, it's in its first and last names. And this bacteria,
Halotolerant.
it's not halotolerant. It's a halophile. It will not grow without salt. So it's an extreme halophile. Not only will it not grow without salt, it needs to have a lot of salt to even start growing. So it is an extreme halophile. It didn't even grow with our 25%. That's theoretical data, remember. It should have, but sometimes it doesn't. What?
I'm looking at it. It might. It's hard to tell if it's like where it actually did a little bit.
Not enough where I'd say if as a general rule, if you're squinting and looking and maybe, then no.
Yeah.
When it does grow, it usually produces this kind of light pink pigment. So the fact that you're not seeing that, it didn't grow as we expected it to. It is a very picky bacteria because we are not the Dead Sea. And it's hard to replicate those conditions in the lab. So that's where extreme halophiles tend to grow, places like the Dead Sea, the Great Salt Lake. It's very hard to mimic those culture conditions in the lab. So if it did not grow, it's not really a reflection on you. I have trouble getting it to grow in the lab. Professor Lopez is usually really good at getting it to grow in the lab, but even he has trouble sometimes. The reason being, this is not its natural environment. It's not meant to grow in the lab. It's meant to grow in really, 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 really salty water. What about Staph aureus? Halotolerant as well, yeah. Because it grows at 0% and any other salt concentration. It doesn't need salt to grow. That's why it can grow at 0%. But it tolerates low amounts. So it can grow until the salt gets at about 10%. Does that make sense? That S. aureus is halotolerant? Why?
Because on the skin.
Their skin, yeah, why? It grows at 0% salt, so it doesn't need salt to grow. But our skin is a little bit salty, right? From the urea and from our secretions make a bit of a salty environment. So Staph aureus lives on the skin. It needs to be able to tolerate the presence of salt. We should also know Staph aureus is halotolerant because why? What did it grow on last week? Mannitol salt agar, right? Absolutely. So we already knew it was halotolerant. This just verified it. Great. I like it when data comes together.
Professor, is there any bacteria over here grow on our tears?
I'm sure Staph aureus would be fine in tears. It's salinity, yeah. I mean, I hope we're not trying to grow things in tears.
I know.
Sad. But yeah, your skin has the remnants of tears. I mean, even when you're not sad, your eyes are tearing.
Yeah.
If you think about bacterial conjunctivitis or pink eye, bacteria can definitely grow. Some bacterial species can grow in the presence of tears. Because otherwise no one would ever get pink eye. Pink eye stinks. Very annoying to deal with. Okay, so the thioglycolate tubes. Have a look at them and try to decide whether your bacteria, how you would classify. Remember the classifications are in your lab manual with pictures. You're going to classify them as either a microaerophile, a facultative anaerobe, an aerobe.
I think
Or a
this
strict anaerobe.
is
So it's on
full
your lab manual towards the back
faculty.
from last week.
something. Get these and then spread around.
Have a look at your thioglycolate
Did you also do? Did we all do E. coli?
I do E. coli. It's E. coli. You're E. coli too, right? Yeah. Yeah.
It's pretty clear.
Yeah, very clear. You're E. coli? Yeah, thank you. Yeah. It's very similar. Huh? Same thing. Yes. How to pronounce it? Faculty anaerobic.
I think that's like facultative, like faculty, like
Yeah,
facultative and anaerobic.
Anaerobic.
Yeah.
Facultative anaerobic.
Kids have a party.
Oh my
So
god, my heart.
we'll say it did grow throughout, but
I guess like facultative.
yes, it does, but try not to
Like
do that.
faculty are people who don't get a place, so facultative means it can work.
Yeah, that's also exactly what I thought when I read.
So a lot of growth at the top and less throughout. I've seen a lot of the same growth patterns, which isn't surprising. So Staph aureus, those who had Staph aureus, how are you classifying it? Facultative anaerobe, so E. coli. Can I borrow yours too? For facultative anaerobes, what you're going to see is a lot of growth at the top and less throughout the tube. And these are two classic examples of what a facultative anaerobe looks like. The reason being, this was yours. This is yours. The reason being comes back to what we're studying this week in lecture with metabolism. So it grows better in the presence of oxygen. So this is growing below the surface and then throughout. So that would be considered a what? If it doesn't grow at the surface but below it.
Oh, micro something.
Micro something. Microaerophile. Right.
Pretty. It's like it looks almost like you traced where I put in.
Yeah, so there's a little bit of agar in the tube, remember? So that's kind of holding the bacteria up and in place. So I see a lot of facultative anaerobes, which is expected. Most of these are E. coli.
E.
Staph aureus.
coli.
B. subtilis is a microaerophile, so it should be growing just below the surface. But sometimes these are a little picky, just below the surface. And that is a facultative anaerobe. You can see more growth at the top. You just didn't get a lot of bacteria in there, but you can still see the pattern. Absolutely. Great. So for this, I just want to get back to why the facultative anaerobe grows better at the top of the thioglycolate broth than it does with it. And it goes back to the idea that there are different pathways to making ATP in the cell. So what are the different pathways for making ATP in the cell that you studied this week and week 4?
It's the pain.
It's what?
No, it's painful studying.
It's not painful. I'm not asking you to memorize the steps. I had to memorize the steps. I don't remember them to this day. That's why I never make students memorize the steps. What I want you to know instead is for each set of reactions, glycolysis, the Krebs cycle, the electron transport chain, what's going in, what's coming out. Okay?
So you don't need a specific ATP.
I need to know the specific ATP, that's what's going out.
Okay.
I don't need to know that first you're going to take glucose and you're going to make it into glucose 6-phosphate and then glucose 3-6-phosphate and then it splits apart. And no, not each single step in the pathway, just generally. So, here, I'll show you. Take a minute and go through this because it is important and we will talk about it again on Tuesday. But I just want to make sure because the thioglycolate tube is a great illustration of how this works. So, to make ATP, you have 3 major portions of a pathway.
Yep.
You start with glycolysis. And glycolysis doesn't need oxygen. It's an anaerobic process. From glycolysis, what's the end products of glycolysis? Anyone remember?
No.
No. I just heard someone say no. That's okay. I like the honesty. So you're taking glucose and you're breaking it apart, glycolysis. One of the end products starts with the letter P. And it's with pyruvate.
Acid.
Pyruvate, yeah. Great.
There's
Pyruvate. You're also making
NADH blood. No blood.
And how many ATPs per glucose molecule coming in?
3.
2 net. Because you make 4 total, and you have to spend 2 to make 2. So the net goal, you make 4, but you spend 2 to get there, you make 2 extra. This is glycolysis. So sugar, glucose comes in, an NAD plus, and some phosphate. And this is what comes out. So that's what I mean, know what goes in, what comes out. 2 pyruvate, 2 NADHs, 2 ATP, 2 ATPs. Then you have what's called the preparatory step. That takes the pyruvate and something called coenzyme A, and it makes something called acetyl-CoA. And in the process, it takes 1 more NAD plus, and does it oxidize or reduce it to NADH? Is NADH oxidized or reduced?
Oxidize. Reduce.
NAD, well you have a 50-50 shot here. So NADH is the reduced form. Reduced forms of molecules carry potential energy.
Got it.
We've taken an electron from pyruvate and we've reduced it to NADH, so it's carrying potential energy to save for later.
So this means it add more.
What?
Like it add more.
No, it's only going to add 1 right now.
Oh yeah,
What?
add 1.
NADH. Then what's the next step in respiration?
Electron.
After you do the prep step, where does the acetyl-CoA go into? What cycle does it start?
Electron trans.
Nope, not
No.
yet, Kim.
Not yet. Krebs cycle.
I heard someone say it. It has many different names. The Krebs cycle. What's another name for it? Citric acid cycle. What's another name for it? There's one more. It's the most tongue-twisting one. The tricarboxylic acid cycle.
Da da da.
So we're just going to call it
Da.
the TCA, tricarboxylic acid cycle.
Then we must have to remember that name.
Yeah. I don't think it's a lot to ask in a college level class that you remember the name of the process. So, what goes in, what comes out, it's a cycle. I am not going to get the amounts right here, but some NADHs are produced. Some ATPs are produced. Actually, 1 ATP per turn of the cycle. And something called FADH2 is produced. something else that can happen. Now you can say it.
Yeah, electron transport.
The electron transport chain.
My favorite.
Okay. So, in aerobic respiration, what is the final electron acceptor?
Oxygen.
Oxygen. So the electron transport chain, let's break down what this means. When you've been making these NADHs and these FADH2s, you've been reducing them. Remember the mnemonic, Leo says grr. Leo the lion says grr. Loss of electrons is oxidation, gain of electrons is reduction. Easy way to remember it. Leo says grr. Leo the lion says grr. It works. So, throughout this process,
So loss of electrons
means something's oxidized. Gain of electrons is when it's reduced. So, when all three processes occur, glycolysis, the prep step, the carboxylic acid cycle, you're making all these electron carriers. Electrons are being held, increasing the potential energy, storing it for later. In the electron transport chain, you need a final electron acceptor. The final electron acceptor is eventually going to take those electrons and convert them into chemical energy into ATP. We call this oxidative phosphorylation. And when aerobic respiration happens, how many ATP per glucose are you going to make? It's a nice little table in your textbook and on the slides. Anyone remember? That's something that's really good to put on a study sheet. 36 ATP per glucose.
Yeah, 36.
What?
Yeah, 36. And now I
All right,
remember.
oh, good.
Now I remember.
Good. So that's respiration. Anaerobic respiration can make anywhere from 3 to 35 ATP per glucose. That's because some portions of the Krebs cycle that make these electron carriers those enzymes don't work in the absence of oxygen. So aerobic respiration is the best, makes the most ATP. Anaerobic respiration uses something besides oxygen as its final electron acceptor. Maybe it's nitrate, maybe it's sulfate, maybe it's carbonate. Any inorganic molecule is its final electron acceptor. And it makes less ATP.
Yep.
We have a third process that can happen here when no final electron acceptor is available. What's it called?
Fermentation.
Yes, fermentation. So, fermentation. How many ATPs per glucose does fermentation help make?
I don't know. 2?
2, right. Because
Oh,
in fermentation, what happens is,
Fermentation.
if you don't have a final electron acceptor, that NADH that's produced will never be able to hand off those electrons to oxygen or another final electron acceptor. And eventually, you'll run out of NAD+ because it's not being recycled. In a chemical reaction, what happens when you run out of a reactant? What happens to the chemical reaction? It stops, right? So the bacteria has a choice. It can die, which is not its optimal choice. It's never the optimal choice. Or it can find a way to regenerate NAD+ to keep glycolysis going. So fermentation is the act of what
Survival
happens
mode.
is NADH gets oxidized while pyruvate gets, something's oxidized, what happens to something
Reduce.
else? Reduced. Right, so pyruvate is going to be reduced and produce things like acids and alcohols. So fermentation only makes 2 ATP per glucose because all it does is keep glycolysis going. And this is called substrate level phosphorylation. You'll see it in the slides that I make the distinction. We'll go over this again. But getting back to the facultative anaerobe, getting back to your thioglycolate terms. For a facultative anaerobe, at the top of the tube where oxygen is present, what kind of process do you think is going on in that cell?
It's aerobic.
Aerobic respiration, yeah. So it's making at the top of the tube 36 ATP per glucose. At the bottom of the tube, where there's no oxygen, what is it likely doing?
Anaerobic.
Absolutely, fermentation. And it's only making 2 ATP per glucose. Where is it going to grow faster, at the top of the tube or the bottom of the tube? Top. And that's why you're seeing better growth at the top of the tube than the bottom. Because at the top of the tube, aerobic respiration is making a whole lot of ATP for that cell. But at the bottom, fermentation takes over, and all you have is glycolysis. So you can't grow as fast, you're not making as much energy. So these things you're seeing in the tubes correlate directly to those biochemical processes. So I noticed something on the exam. I just want to take a minute to talk about. And it kind of ties into what we just talked about here. Many of you, some of you did, but a lot more of you than I expected to didn't bring a study sheet with you. A note sheet. You just opted not to. And I really want you to get into the habit of doing that. First off, because it helps you keep up with your studying in the class. And second, there's a lot of things to know, especially with unit studies. So what I'm going to encourage is, instead of being able to use the skeletal notes on the review quizzes during the week, I'm going to encourage you to bring a small index card with you. And if you need index cards, I have plenty in my office, of notes for the next review quiz. So on Tuesday, we're going to review metabolism. Just make an index card or cut a piece of paper smaller than usual and bring a couple of notes you think might help you. That's all you're going to be allowed to do. And when you go into your group, you're going to compare your notes with your classmates and see what helps you and what doesn't. And then that will help you build your study sheet for the next exam, which is going to be after spring break, after we finish Unit 6. This is a jam-packed unit. We do metabolism and everything that involves, as well as bacterial genetics and molecular biology tools. So there's a lot in this. So I really want to encourage you to maybe try and do that for the next 3 review quizzes. And let's see if we can get it up to 100% using well-planned, well-thought-out study sheets for the exam. And we'll see if it helps.
I have a question. So you said one face of the index card?
Even 2 sides of an index card.
Okay.
I just, instead of a whole sheet of paper, let's just keep it manageable. And that way, if you're stressed right before the test and you're like, oh no, I didn't make a study sheet, you could take those 3 index cards and just stick them on a piece of paper and you're done. Give it a try. If it doesn't work, we don't have to do it again.
So only index card, because you also mentioned that index card and couple note. So
You make the notes on the index card.
oh, okay, I got it.
Okay, so what you're going to do now is take a little bit of alcohol and remove the labels from the test tubes, put them in the discard bin, and you can just get rid of the Petri dishes. I'm going to erase the board, and
We're
then
gonna
I'm
clean?
going to take out your hand washing, so you can look at that. Let's see how we did at washing our hands. So what you're going to do with those plates when you're ready is you are going to count the number of colonies on the before and after plates, and we're going to report them up here. Don't worry about light. If you found my whole spiel about respiration and fermentation confusing,
We
just
erased
know
all?
this is a rare instance where I made a video. It's on the week 4 video library of me going through exactly this. Again, what are the inputs, what are the outputs?
I'm sorry, you erased them all.
I did, yes. It's okay, I'm not going to quiz you on that. But as I was saying, there's a whole video of me going through it again And you're just going to record the number of colonies on the before plate and the after plate. Oh no, I thought I put things in that warm basket. I did not. Okay, so count your colonies and put the counts up on the board.
Yeah, the timetable is like 1 hour. Oh my god, it's still.
Yeah.
I'm scared.
Colonies, count big colonies, count fuzzy colonies.
I'm so scared. Oh my god, it has so many colors. Ooh. Oh, it's so clean. Oh, this after. Wow. You're also different color than. Yeah, she only like white and then open yellow.
Any numbers there? Just write them in the same box. If you didn't do plays and you were out
There were like 27 in this one and just one.
But it's really interesting. But honestly, even if you wash your hand, you still have a lot of. Interesting. Wow. Now I'm scared. So basically, you would walk out and then have some little more folding. Oh my god. But it's beautiful.
Well, I don't have to, but I want to look at what they look like.
And there's this. I have a lot of inoculations to do today, so I'm trying to get some in whenever I can. Betadine. Pretty good. Almost 75% reduction, 50% reduction before and after. Betadine is a surgical soap. It's used by surgeons when they scrub into surgeries and things like that. It's a hospital grade soap, so I'm not surprised to see that level of activity from that ability. Dial soap is pretty good as well, except for this one where you end up with more. I don't know why the dial soap. A lot of times, I suspect that. So when you pump, people are recontaminating their hands and then washing, so they add some more bacteria before they take them away. So what I've been doing is occasionally going over, because my other class is actually the same thing, just cleaning off the nozzle on the dial, so it appears to be problematic. We'll just let it sit there for a minute.
And sanitizer.
And the hand sanitizer, woo!
I love hand sanitizer.
That's 100% reduction.
Wow.
Hand sanitizer is a CDC recommended and approved way to take bacteria off hands only when they're not visibly soiled. If your hands are visibly dirty, hand sanitizer doesn't work as well because the alcohol doesn't get in contact with the actual microbes. It gets in contact with the dirt, which interferes with its activity. So if you had to choose personally, what would you want to wash your hands with?
Soap.
Someone said hand sanitizer. Someone said soap. Which soap? Which soap did the best?
Dial soap.
Well, it depends which data you look at. 27 to 1 is pretty dramatic, but 25 to 40 doubles it. So, I don't know. Whereas betadine had consistent, but betadine has issues if you're allergic to iodine. You shouldn't use betadine because it's all iodine. That's the active ingredient. Whereas dial soap uses something similar to what's in Lysol, but is gentler for the skin. It's benzalkonium chloride, which is an ammonium compound that interferes with microbial cell walls.
Also, different hand soap. Different, right?
Yeah, I mean, we use the dial soap because it has that extra antibacterial ingredient, and we're working with a lot of bacteria in the lab. But like your regular like Bath and Body Works soap or Dove has no antimicrobial ingredient, but it is still better than water alone. But if you're camping and have access to clean water and no soap with you, rinsing your hands and rubbing them together didn't do a terrible job. If you have no other alternative, at least rinse your hands. Now, sometimes people are alarmed by this because they get some interesting looking colonies on the plate, different colors, different levels of fuzziness. That's okay. Also, the number of bacteria on the plate isn't really the most important thing. We all have bacteria living on our hands. We need that bacteria living on our hands. Our skin would not be healthy without it. The bacteria on our hands produce acidity, which helps protect us from microbes that don't like acidity. They also take up room and nutrients so that bad bacteria can't come in. Too crowded. You don't want to completely sterilize your hands. What's most important is the type of bacteria that is left on those plates after hand washing. So let's say you're walking down the hall, Kingsborough, and you grab a doorknob, and right before you, someone with some sort of bacterial infection coughed on their hands and touched the doorknob. You have so many bacteria already on your hands that bacteria is just going to kind of stick on the surface. But before you go eat your lunch in the cafeteria, you're going to wash your hands and you're going to rinse that bacteria away. So that small amount that you just picked up transiently or temporarily from the doorknob is going to get washed away before you have time to ingest it or it's time to extract it. Yeah?
Bacteria, like let's say you want to travel in space or something, could you engineer
Could you? Yes, probably. Should you? Yeah, we genetically engineer bacteria all the time to have specific traits that we want to study or we need. We'll talk about that all in week 6, different ways we can alter the genetics of bacteria. We use bacteria as tiny little factories to produce medicines, to produce things we need industrially. When you buy rubbing alcohol at the store, that is produced by bacteria in large vats in fermentation tanks in a factory. The vaccines we take are sometimes from genetically modified bacteria that are genetically modified to produce a specific portion of something that can cause us harm and then is purified into a vaccine. So we can definitely genetically engineer bacteria to have specific traits. Just give me a minute. But the question is, could you and do you need to? And the answer to that is a lot more. Usually, probably not. The reason is there is so much we still don't know about our microbiomes. This is unexplored territory that lives right inside of us. That we don't know the consequences of certain modifications to our microbiome. We know that babies born by C-section have different microbes than babies born vaginally. And sometimes that might predispose them to diseases later in life, like diabetes. However, we don't know why. So messing with that without knowing the why or how it works can be really dangerous. So the question is, should we know? You probably also don't need to. Just because you're microbes, you're their ideal environment. So as long as you're there, no matter where you are their ideal environment. Your diet can change that. Like if you've ever taken antibiotics and had your doctor recommend, have some yogurt. That's because the antibiotic is going to kill off a lot of your intestinal good bacteria as well, and you want to try and repopulate it with something helpful. So there are ways we try and modify it, but we don't understand enough about it yet to deliberately force that issue. Yeah.
So I wash my hand a lot and I have eczema. So does this come from another bacteria or?
It comes from washing your hands too much. I have eczema too.
Oh,
yeah. So you're removing your skin barrier,
oh.
the acidity and the sebum on your skin that's protective, and making a less hospitable environment for your good bacteria. And that makes your skin dry out and crack in your immune system. Eczema is largely autoimmune, so your immune system starts attacking.
So eczema is not a bacteria cause, but it's more like cells.
It's not like bacteria. Usually it's treated by immune modifiers and steroids.
Because all my
suppress the immune reaction.
all my life, I thought it's like a caused by bacteria.
No, I'm the opposite. As I go to wash my hands yet again, because they have chalk dust all over them. Something I probably shouldn't be doing. But yeah, it comes from usually genetic factors, and then washing your hands too much can make it worse.
But it's interesting. I wash my hand a lot when I was in my country. It's very worse. But when I moved here, I still wash my hand exactly the same amount, but it's less.
Different soaps, I don't know. Not a dermatologist, but oh my goodness, that is really simple.
Yeah, that's why I did.
Okay, so any questions about this? Yeah, ask.
Can we identify which ones
You could. What are some ways we could we're not going to. But if we wanted to, what's one thing we could do to those bacteria on the plate to identify them? Place them into 2 broad groups, maybe.
do like a different?
I heard it. Gram stain. We could gram stain them. We could plate them on mannitol salt agar and see if they ferment mannitol. We could plate them on conky agar and see if they're gram negative, and if they are, do they ferment lactose? Because might they come from, say, someone's intestines? We could do a lot to characterize them. We're not going to though, because we don't know what's growing on our hands, and we don't want to potentially introduce something disease-causing that could be harmful. Because while it's on your hands, it's in low enough amounts not to make you sick. But when we grow it up in a culture,
Oh my
it
God.
can become more dangerous. That's why we keep culture volumes as little as possible, and we do all this sterile techniques. So yeah, it's amplification. So we don't want to. You're exposed to bacteria all the time. However, there's something called an infectious dose. Many bacteria and viruses, for that matter, have different infectious doses. That's the dose it'll take to make most people ill. So if you're exposed to a little bacteria, your immune system will handle it. But something growing on a plate probably has trillions of cells per colony. We don't want to potentially introduce that. So we keep the plates closed and we get rid of them.
Do you know what these little yellow ones are?
Just by looking, I'm not that good. However, yellow colonies usually, remember I told you about the name of the bacteria? AU, gold, Staph aureus. Wouldn't be surprised if that was on everyone's hand because most adults carry it. So yeah.
But even though we don't have to stay, but it still have a different color.
It happens. Okay, so discard your plates and we're going to move on with starting our unknown analysis. Very exciting. So you can just discard those on your erase the board and I'll take you through what's going to happen over the next couple of weeks. So today you should be taking your week 2, right? Yeah, week 2 pictures of the Winograd. Remember, take pictures of all 4 columns that our class made, as well as all 4 of the columns from the previous year. Make sure you label them.
Also, I thought this week is like week 3 or week 2.
We are week 4 in lecture. This is week 5 in
No,
lab.
I mean the-
Oh, no. Week 0 was the week we
Oh,
made
week
them.
0.
Start week 0, yeah. So if your numbering is off, it's fine as long as we're consistent
Because,
with it.
yeah, I didn't have
Okay. So, today in these exercises, we're about to do on carbohydrate fermentation. We start the series of experiments for your first unknown report. So this is a lab report where 75% of your grade, not 75%, oh my God, 75 points on your grade, 7.5%. Big difference. Sorry about that. I didn't mean to scare anyone. What we're going to be setting up today is several tests: the phenol red test, the oxidative fermentative glucose test, methyl red, Wilkes Proskauer, and citrate tests on 4 different organisms: E. coli, Enterobacter aurogenes, Proteus vulgaris, and Pseudomonas aeruginosa. These tests you're also going to be setting up on your own in about 2 weeks, the week after spring break, essentially. As your unknown. So what happens is these are our controls. We're going to set up carbohydrate controls this week, analyze them next week, collect the data. Next week, we're also going to set up protein metabolism tests on these same 4 organisms that are going to be our controls for additional tests you're going to run. When we come back from spring break, we analyze our protein metabolism tests and we make a chart with all the control results. This is what you're going to compare your results on your unknown to identify your unknown. That week, I'm going to hand you a little culture tube just like this, but instead of having the name of the species on it, it's going to have a number. Your responsibility independently, this is an individual exercise, will be to set up all these tests on your own. You are allowed to use your lab manual, of course. However, I will not be telling you how many tubes you need or what you need to do, and you won't be talking to each other to do it too. You have to set these up on your own. I will, of course, stop you if you are doing anything dangerous, but otherwise, this is an independent exercise. We're going to practice setting them up today and next week. So it's really important that you're here, and if you're not here, then you can do the lab manual in order to keep up. So last week, you are going to analyze all these results, and you're going to write a short report about how you identified what your unknown bacteria is. And it's going to be one of these 4 species. So, writing the report, I'll take you through it after spring break. It's a very formulaic report. I'm not asking you to write an abstract, an introduction, materials, and methods. It's not that type of lab report. It's modeled after a clinical lab report. So you're going to fill out a chart that's going to tell me what your observations were, what the result of that test based on your observation is, how the test works, and what organisms have that same result. Then for the discussion part of the report, the first part of the discussion is what are the 4 possible bacteria that you could have gotten. Based on your carbohydrate fermentation tubes, were you able to eliminate any bacteria? Based on the next test, did you eliminate anything else? And you're going to go through your test until you're left with one possibility as an unknown, and that's how you're going to identify it. All in all, if you do it right after class, it should take you no more than 2 hours to write. It is not a very intense lab report, and it's very, this is what I want, this is how you write it, and I'll take you through it again. But my point is, for the next couple of weeks, it's very important to get this information. Because online, I have theoretical results. As we know, sometimes the theoretical results do not match what we see in lab. We are going by what we see in lab. Because that's how that bacteria behaved that day. If we try to use the theoretical results to this, it might not work. I don't know how it's going to work. So just keep that in mind as we go through this today. I'm going to assign each row an organism, and you are going to set up organisms, the tests for your organism in that row. Okay? So if I give your row E. coli, you're going to set up all these tests for E. coli per row. Because we don't have enough media, I think, for everyone to do it. Do it by pair, there are 16, I need 18. You know what? We can do it by pair. So you'll pair up. You'll pair up and then do it in duplicate per row. Does that make sense? It'll make more sense as we go on. So let's go in random order today. So row 1, you'll do Enterobacter aerogenes. Row 2, you'll do E. coli. Row 3, you'll do Pseudomonas. And row 4, you'll do Proteus vulgaris.
Is it row 4?
Proteus vulgaris.
Proteus.
So the first 2 and the last 2 pair up together. That way everyone gets a chance to practice setting these up. Because sometimes we only have 2 tubes in the test. Sometimes there are 3. So make sure you get practice doing these. They're all aseptic transfers, so it's nothing really new technique-wise, but I'll take you through what the medium is and how it works. So, we're in the week 5 lab manual now, and we're going to go in order today. So, the first test we're going to set up is something called fermentation tubes for carbohydrate fermentation. And for this test, I have 3 different media that you're going to need to inoculate.
Also, Professor, because you said that after spring break we have to identify the bacteria by our own, but now we only have the chance to do 1 to observe. So
Yeah, and then we're going to share the data throughout the class to make a table to compare. You don't need to set up every bacteria to see the results. Yes, Devin. No, we're not using Klebsiella. No, these are the bacteria. Enterobacter, aerogenes, E. coli, Proteus vulgaris, and other bacteria. It doesn't list Klebsiella, so I'm not sure where that's coming from. But we're doing these tests on these bacteria only. So, the fermentation tubes. PR lactose, and PR mannitol. When you come up to take your tubes, you're going to take 1 tube of each. Per pair, you're going to take 1 glucose tube, 1 lactose tube, 1 mannitol tube. This is an instance where you need to label it before you walk away, because otherwise, you walk away, the glucose tube looks exactly like the lactose tube, looks exactly like the mannitol tube. You don't want to mix these up because each bacteria might ferment those 3 sugars in different ways. So it's really important to keep them organized and label everything properly. Again, you're going to label the name of the media, the organism name, the date, and your initials. Just one initial, that's fine. You don't have to try and squeeze everyone's initials on there. So, the way this test works, you have these tubes, and the tube contains broth. The broth contains 0.5% of a sugar, either glucose, lactose, or mannitol. It also contains our favorite pH indicator, phenol red. Phenol red, remember, is red at what pH? 7, right? And it turns yellow at what pH? 6.8. It's very sensitive. It will change color at very small changes in pH. In this tube, you will also find an upside-down test tube filled to the top with media. This is called a Durham tube. The job of the Durham tube is to trap gas. So, what we're testing here is the ability of the bacteria to ferment different sugar sources. So, if the bacteria ferments the sugar, it's going to produce acid as a result. Acid is going to do what to the pH? It's going to change it, but it's going to drop it. Right, here's that. Drop it. Yes, make it more acidic, drops the pH, turning it to yellow. Okay. If it does not ferment the sugar, yeah, it's going to remain the same. So, no acid. What color will the tube remain? Red. So that's part one. We're going to look next week at the color of the tube and see if it's red or if it's yellow. If it's red, it's going to be negative for acid production, meaning it does not ferment that sugar. If it's yellow, it's positive for acid production, and it does ferment that sugar. So that brings me to the Durham tube. There are different types of fermentation, and there's different products of fermentation. Sometimes you produce acids. The types of acids can be important too. Sometimes you produce alcohols. Sometimes you produce gas. All these different little nuances of fermentation, all these different little ways to ferment a sugar, all these different byproducts. So remember, and you're reducing it to something else, acids, alcohol, gas, etc. All in the hope to take NADH and oxidize it to NAD+. So we're looking at what type of fermentation by looking at the byproducts of pyruvate reduction. We want to also measure not only is acid produced, but is gas produced as well. So that's why we have the Durham tube in here. What you'll notice is the Durham tube is filled to the top right now. There's no room for anything else in that Durham tube. It's a little upside down glass test tube in here, and it's filled to the top. When you get more media, just notice that your Durham tube is full to the top. It might not be next. What happens is some bacteria, not all, but when they ferment the sugar, will not only produce acid, which will drop the pH, but will also produce gas. The Durham tube will trap that gas. And what will happen is you'll get a bubble in the Durham tube. So the Durham tube will no longer be completely full. There'll be a little space at the top with some gas in it, with some air. and you see a bubble in the germ tube, that's positive for acid, positive for gas. And if you see yellow with no bubble, which is a possibility, it's positive for acid, negative for gas. In both positive for acid situations, that means it ferments the sugar. You're just looking for what does it produce as a byproduct. Does it only produce acid, or bless you, or does it produce acid and gas? You'll never have an instance where you have a bubble and red. That doesn't happen. There's no type of fermentation that only produces gas. So what you're going to do now with your partner is, between the two of you, you're going to take one glucose, one lactose, one mannitol tube, and you're going to inoculate it with your bacteria, which I'm leaving here. Make sure you take the right species, and make sure you label it with glucose, lactose, mannitol before you walk away. If you need a refresher on how to do an aseptic transfer, I'm going to wash my hands real quick, and I have lots of aseptic transfers to do. So, I'll wash my
What is this called?
For some of them, I only have 3 right now because I'm still inoculating. So, actually, for all of them, you're going to have 3 because I need to grab them. Keep them contaminated. I'm sorry. And these are Eosa and Bulgaria.
We only need 3.
Okay, yeah, I got one.
I don't even read it because the pain, I think the pain you gave me is a little small. So I know glucose, lactose, let me see.
So I start off.
And our name? Yeah. You are in
MOS. Usually, I usually include my middle name.
and 7th of April.
April 3rd, today.
And the bacteria is the Bulgaris, P. Bulgaris.
So you're taking the bacteria from the culture tube and you're putting it into the fermentation. You're going to incubate everything in your drawer over the week. If you feel your loop is getting a little hot, you can feel free to take a paper towel and wrap it around the handle. There's glucose, lactose, mannitol. You have to look at what it says on the chart. And I'm doing lots of aseptic transfers. I should change
Yeah.
I guess with this.
Yeah, take some so you have the picture.
I don't want to.
make sure your left partner can see you. So I'm doing it right now. I'm going to take bacteria from this tube, which is the culture containing tube. It's nice and turbid, and I'm going to put it in this. Seven. Are you going to be setting one up? Make sure everyone gets this. You're going to have to do it on your own.
Because there's other.
Yeah, so the next one, they also have another bottle. I'm not sure.
I think we'll read the instructions first.
Yeah, that's why.
I'm trying to figure out where this is. Get ahead of it.
My brain just shut down after the screen test. So I'm like, now this level is I can function normally until I get a full sleep tonight.
Yeah, we did all three.
If you're waiting, the next test we're going to be doing is oxidative fermentation glucose, which is the next in the lab.
I passed it everything, but I think
She's really flexible. I've turned things in late with her, and she never takes it.
yeah. Finish that now. It really, really is.
You're going to need a culture tube. Sorry, you're going to need it for the rest of the class, so you might as well hang on to it. You don't have to bring them right back up this time. Because you're all doing the same organism over and over again. So I'll leave you walk.
You always keep asking us, where's my tube, where's my tube? And I'm like, now I'm like,
I don't need to be eating. I just have to make sure everything's accounted for. It's not a security risk. You can have two bacteria running around your hands.
then I know that I got so stressed. I get so stressed, like, then I also gain weight from my day. It's messed up my day. Right now.
I mean, I always have acne. It's just like genetic, but like it's been especially bad lately for me too. It's like, ugh.
Yeah.
Okay, it looks like we're ready for the next one. So the next test is what we call oxidative fermentation. Research, and again, if you miss any of this, it's in the review slides. There are videos about each test online, so you have lots of resources to look. Keep in mind the slides that are on the right space contain theoretical data. So that's that's how it should work out next week, but as we know, sometimes things don't work out the way we want them to. So, on the next page, you okay? So, our last test looked at fermentation. Just move these to the side so we don't get confused. Next test, the previous test we looked at, does it ferment a specific sugar? Either glucose, lactose, or mannose. That's what you're going to find out next week. This next test, oxidative fermentative use of glucose, is going to ask the question, does it use respiration? In this case, aerobic, or fermentation to break down glucose. So, what we have are these tubes marked OF glucose. And you can notice right away they're a different color than what we've seen previously. This broth contains glucose, thus the name. So we have our tube, and it contains glucose as the carbon source. It also contains a pH indicator called bromothymol blue. Bromothymol blue is green at pH 7, so neutral pH. When it gets acidic, it's going to turn yellow. So we're checking to see if glucose metabolism is going to produce acid. If glucose is fermented anaerobically, it doesn't need oxygen to do that. So each pair is going to take 2 tubes of this bromothymol blue, We're going to set up 2. One without oil on top, and the second, you're going to layer after you inoculate. So you inoculate first, you add the bacteria first, and then you're going to layer it with a small amount of mineral oil, sterile mineral oil, right on top. The job of the mineral oil is to keep out oxygen. So, basically, we're testing the ability to use glucose under oxygen conditions and non-oxygen. If the bacteria uses glucose fermentatively, meaning only via fermentation, you're going to see acid produced in both tubes. Because fermentation doesn't care if there's oxygen or not. It's going to take place regardless. So, fermentative use of glucose means yellow in both tubes. However, if the bacteria only metabolizes glucose via respiration, aerobic respiration requires oxygen. So which tube do you think is going to be yellow if the bacteria uses glucose oxidatively only?
No, Manu.
The first one, the one without oil that has access to oxygen, absolutely. So oxidative use, yellow in no oil tube only. So to set this up, each pair takes 2 tubes. Each lab partner inoculates a tube. Then in one of the tubes that you're going to label no oil, sticky on here, come off with alcohol if it didn't already. Just over it. It's fine. Your marker is going to be a lot higher. But I'm going to just encourage you to work at your bench because once you crowd it over here, you're waiting in on the same speed. Okay? Like working with that order because you need to have a space. Because they don't want anyone accidentally elbowing or flicking bacteria.
Let me label. You want to label this?
Yeah.
It's OF glucose. That's what you should label it. Because remember, it might not be green next week. So it'll look just like all the other yellow tubes you have, and you might not be able to read your result. So always write the name of the media on it.
Oxidative.
So only half of you need oil. Yeah, so you can just borrow from someone else, but yeah. Remember, every person is not adding oil onto their media. One dropper full. So you want like a layer. The job is to keep out the oxygen. So there's no too much, but if you add too little, it won't completely cover the surface.
No, we're gonna. I think we're gonna.
Put first. Yeah, and then I will do this. Sure.
Yeah, you don't need the oil anymore, so when you're done with it, you can just bring it right back up here. Okay. So the next tests we're going to be setting up are the methyl red
You want it.
and
You want it full
scour
layer
tests.
so that.
And we set those up together because they use the same medium. So this medium looks far less exciting than the others we've used. This is just a broth containing glucose. acid fermentation. You'll see 2,3-butane-diol fermentation. These are all classified by what those end products are. So this is another way to classify what type of fermentation is going on in your bacteria. And I hope by now you appreciate that all these different patterns of fermentation are going to help you identify your bacteria because they're unique throughout these different species. So in addition to things like gram staining, metabolic testing also helps identify a species of bacteria. So for the methyl red test, this is a test for something called mixed acid fermentation. So glucose is present in the broth. If it's fermented, these bacteria capable of mixed acid fermentation are going to produce formic, lactic, acetic, and succinic acids. What do you think that amount of acid is going to do to the pH of the media? It's going to drop it a lot. It's going to be really acidic. I heard several people say, very acidic. That's right. So, phenol red. Phenol red is the pH indicator we use so much. pH 7 is red. It changes at pH 6.8. A very slight change in pH. So, phenol red can't distinguish the difference between a little acid and a lot of acid. It just changes with a little bit of acid. So we need to use a less sensitive pH indicator to see if it's mixed acid fermentation or just something like lactic acid fermentation. So what we do is we let the bacteria grow in the tube, and next week we're going to add a pH indicator called methyl red. Methyl red is yellow at pH 7 and doesn't turn color to red until pH 4.4. So you need almost 1,000-fold increase in acidity to see a color change with methyl red. It's not sensitive. So it's going to take a lot to change that color. So if next week you add your methyl red and it's yellow, it's negative for mixed acid fermentation. If you see a change to red, that means it's positive for mixed acid fermentation. We're looking at what type of fermentation. Most ProScour uses the same media, so we're looking at glucose fermentation, but we're looking for a different type of fermentation. In this type of fermentation, we're looking to see if the bacteria produce something called 2,3-butane-diol and ethanol. We call this alcoholic fermentation. And in this reaction, there's an intermediate called acetone on the way to making 2,3-butane-diol. Next week, we're going to try and detect the presence of acetone by adding something called Barrett's reagents. You're going to add equal amounts of something called Barrett's A and Barrett's B. If 15 minutes after adding Barrett's A and B, it turns pink, that's positive for alcoholic fermentation. If it stays yellow, that's going to be negative, meaning no acetone is present. And as you study and as you write your lab report for this one, these little blurbs, these little tiny paragraphs at the beginning of each part of the lab manual contain everything I just said and everything you need to know to write your report. So those would be a really helpful guide as well. So for now, you're going to label one tube MR, one tube BP, just because if you accidentally add methyl red to the same tube as your BP test, it's not going to work. So I always find it helpful, even though you're setting up the tubes in the exact same way, label one MR and one BP and you won't mix them up next week. But labeled your organism name, your initial date, MR, and VT. So you need 2 tubes of this, just a regular aseptic transfer.
You are in R or in P. Next one, we're gonna put in as 11.
Okay, so here's another one. Take the label off it first if you labeled it. It's interesting that you didn't notice it was empty as you were labeling. Be all about the definitely. We should all be feeling like pros in aseptic transfer by now. Very close to the sterilizer. I don't want it to catch fire. Do you have blue or purple? Yes, there is something that might turn blue on you, but not purple. There's very little purple in these tests besides the Ramstein. There is purple in a test we'll do next week, but it's more like a blackish purple. Sorry, I'll work on that. Yep. Yeah. So we do setup and then the next week you do analysis. The independent part is I'm gonna leave the media up there and that's it. And you do. And then you incubate it in your drawer exactly as you're doing now, and next week or the week after you set it up, you're gonna come in and do the analysis that we're doing next week. Yeah. Each of these take 2 labs. So if you miss it, there's no real way to make it up.
So today, basically, we're just gonna be like practice, like how and observe.
Next week you're gonna observe. There's
Yeah.
nothing to observe yet.
Yeah, observe like this process, but like the real analysis and for the lab report is like when we do the one that has no name. Like we have to. Okay, got it.
Exactly. These are the controls. You can't identify your unknown unless you have something to compare it to.
Okay. Yeah.
So this is what you're going to compare.
Okay.
Good questions. I'm going to post the report So the last test we're going to set up for carbohydrate fermentation is the citrate test. And this one's a little different. We need more. Unfortunately, our lab technician just needs some more. But Angela, you are not alone. We get empty tubes and we just roll with it. So I just need to get 3 more of these and then I will tell you what it's all about. This is one of my favorite tests. Shona and Olivia asked me if anything was blue or purple. This one turns a beautiful blue when it's colored. It's one of my favorites. This is the citrate test, and it's found on the next pages. In order, yes, we're going in order today. Citrate. Where do we see citrate in those weak 4 reactions? Where does it come up? It's the input of something.
The methylene?
Citric acid cycle. It's in the name and everything. The citric acid cycle. So citrate is the first step in the citric acid cycle, otherwise known as the Krebs cycle, otherwise known as the tricarboxylic acid. Citrate. Some bacteria can use citrate as their only carbon source. They don't need sugars. Citrate is sufficient because they have an enzyme called citrate permease. And citrate permease is an enzyme that helps the bacteria get citrate across its cell wall and cell membrane so it can be used as a carbon source. When that occurs, the citrate is converted somewhat counterintuitively into pyruvate, which then goes through the preparatory step and back into the citric acid cycle. Do not ask me why it can't directly go into the citric acid cycle. I don't remember that much of biochemistry, but I'm sure there is a good reason. So it gets converted to pyruvate. There's 1 carbon difference between citrate and pyruvate. So what is also made as a byproduct is carbon dioxide. That extra carbon has to go somewhere. We look for the presence of the enzyme citrate permease by using this media right here. This media is called Simmons citrate agar. And it contains, well, first off, and I'll get very excited, this is a defined media. This is the first one we've used. It's a defined media. It contains citrate as its only carbon source. There's no sugar in here. We know the exact chemical definition of this media. Citrate is its only carbon source. You'll also notice that it is this pretty green color in the fall semester. I was like, ooh, it's marketed perfectly for Wicked. It starts out alpha green. So this pH indicator we just saw a couple of minutes ago. The pH indicator in here is bromophenol blue. So Simmons citrate agar is a defined media. It contains citrate, and it contains the pH indicator bromophenol blue. It contains one more thing you need to know about, and that's a whole lot of sodium. Sodium ion is 2 positive or positive? Somebody was taking chemistry recently. Oh well, doesn't matter. There's a lot of sodium in here. For the purposes of this, we don't need to know exactly what the charge on sodium is. Okay, so what we're going to do is this is an agar slant. You only need 1 per pair. But I have another. I will demonstrate, since we haven't done an agar slant in a long time, how to inoculate an agar slant in the aircraft. So what happens? If the bacteria can get citrate into its cell and use it for metabolism through the enzyme citrate permease, that's what we're testing for. It's either either going to be positive for citrate permease or negative for citrate permease. It's going to produce pyruvate as a result and carbon dioxide. That carbon dioxide is going to combine with the sodium in the medium. Yeah, it doesn't say which part is there. When we get NaCO2, this is known as bicarbonate. More specifically, sodium bicarbonate. Anyone here bake? What's sodium bicarbonate? Baking soda. What does baking soda do to the pH of things? What? Other way. Raises it, yeah. It makes it more alkaline. So, bromothymol blue, we know under acidic conditions is what color?
Yellow.
Yellow, right. It's the same indicator as the OF glucose test. Neutral
Green.
is green, it's emerald or alpha green, depending on if you like a wicked movie or not. When the pH rises due to the presence of the bicarbonate ion, that's alkaline, it's going to be a beautiful sapphire blue. It's going to change from green to blue if the organism is positive for citrate permease. So positive is blue, negative it stays green. So, back, which you need it. You're going to take 1 tube per pair of the citrate. And I'm just going to show you real quick how to inoculate this. So I have my culture tube in one hand and my citrate tube in the other. You can inoculate it straight up and down. Just make sure you get the loop on the agar. I prefer to do it this way, just holding it perpendicular because it's a little easier for me to get the surface. You do what's comfortable for you. Sterilize my loop for 10 seconds. Let it cool. Just a little bit. Take off the lid. Remember, nothing rests on the bench. I like to touch the inside of the tube to cool it down a little faster. A little bit of my bacteria. And then using the pressure, equivalent to tickling a feather on something, I'm just going to put my bacteria on the surface of the agar. Making sure I get it on there. And then I'm going to sterilize my loop again, and that's it. So go ahead, get started, and again, everything remains in your drawer. After this, we have one more set of experiments to do. So I'm going to press
Citrate.
and then for the next exercise, we're going to and it does sound great, because I think it's CO2. It's NaCl2, you're right, absolutely. Okay, let's correct that. Thank you. Like I said, I have the week where I don't really, the whole change in schedule really messes with me a lot of ways. Yeah, it's wet. Citrate. CIT, I like to label this too, but you can write out the whole word citrate if you want.
gonna put some.
Yeah. 2 seconds exactly the same.
Everything is going to get incubated in your tumor. When you're done, please bring your culture back up because we're done with it. That is not meant for skin. Be nice to your skin. You might need to wrap because we're going to set up some solutions. Sorry, you're not done with solutions yet. But this is a great activity. As I said in my intro to you, this is my favorite. We're about to do. It might be your favorite. It might not be. That's okay. I have enough enthusiasm for everyone. So we did get the sample from class week of clay. This activity is the isolation of antibiotic-resistant bacteria from soil. As you may have heard on the news or in your doctor's office, there's a crisis in the world where we're running out of effective antibiotics to treat bacterial illnesses. Bacteria face a lot of selective pressure. They can evolve and adapt very quickly to resist a lot of the antibiotics we use. Therefore, drug companies and scientists are always looking out for new antimicrobials to help cure these bacterial diseases. Because without them, simple things like cuts and scrapes and ingrown hairs can become deadly. So, the antibiotics that we come to rely on to treat strep throat, tonsillitis, and pneumonia are really important for our survival. So, one of the ways drug companies and scientists look for new antibiotics is by looking in the soil. Soil contains classes of bacteria. It's not safe to stand in the eye of the
Oh.
Everyone has.
You said the scientists are finding bacteria that are resistant to antibiotics, like superorganisms.
Not superorganisms. These are just regular organisms. You might hear them called superbugs. I've heard that in the news. Superbugs. It's a catchy thing. There's nothing really super about them. These are just normal bacteria that have picked up a trait that helps them resist the drug we use. For example, if something's penicillin resistant, it can produce an enzyme that destroys the penicillin. So it can't work. Something called beta-lactamase. It's an enzyme that digests penicillin. Sometimes bacteria will grow stronger cell walls in order to resist an antibiotic that works against the cell wall. Sometimes bacteria will make extra enzymes. Remember, penicillin targets transpeptidase. So the bacteria will produce more transpeptidase so that all the penicillin can't possibly stop every copy of the enzyme. So bacteria have a lot of different ways of resisting antibiotics. You might hear them called in the news MRSA, methicillin-resistant Staph aureus, Versa, which is even scarier, vancomycin-resistant Staph aureus. That's something you usually find only in hospitals, thankfully, for now. There's also, you might hear MDR-TB, so multi-drug resistant tuberculosis. Then there's something called XDR-TB, which is extremely drug resistant tuberculosis. You might have heard reports in the news, particularly if you have community around nursing homes fungus called Candida auris, which is very antifungal resistant and in fact immunocompromised people with things like catheters and IV lines, ports for chemotherapy and things like that. It forms biofilms and can infect those really easily, and it's very resistant to antibiotics. So these are all things that motivate scientists to try and discover new ones that we don't have widespread resistance to yet. In the soil, there is a class of bacteria called actinomyces. These are filamentous bacteria, which means they don't reproduce by binary fission, but instead they use fragmentation, which we talked about previously. This class of bacteria is very well known for producing antibiotics. And you may think, wow, that's weird. I always thought of antibiotics as something that kills bacteria, not something that bacteria make. Why would bacteria want to make antibiotics? Kill other bacteria, yeah. The microbial world is brutal. If I kill my neighbor, if I kill that bacteria next to me who's eating all the stuff, I can eat the stuff. So the antibiotics are produced as a survival mechanism of the bacteria so it can get more food and resources. And the definition of an antibiotic is something made by a living thing that kills or stops the growth of another living thing. We make an antibiotic. In our sweat and in our tears, there's an enzyme called lysozyme. Lysozyme digests peptidoglycan. So we're a living thing. Lysozyme inhibits the growth or kills another living thing. That's our natural antibiotic. Unfortunately, it's not very potent, but it does work somewhat to keep, our skin and our eyes healthy. And it's also in our saliva as well, to different concentrations. So we're going to look for antibiotics, but first we're going to isolate actinomyces from the soil. And I have 2 soil samples. Mass was nice enough to bring in a sample of clay from the Poly Lab, and they're going to try it. They'll do that one. If you want to try it as well as here, but I'm going to encourage people to also use the KCC farm soil, because soil is particularly rich in actinomyces. Clay can also be rich in actinomyces in soil. I don't know the processing on this, so we're going to see what happens. It'll be exciting. I think it'll be interesting. But there can be billions of actinomyces in a single gram of soil. So, much like we did for our viable plate counts, we need to dilute the soil in order to get colonies on a plate that we can later select and purify. So the goal of today is to dilute and culture the bacteria so that we eventually get single colonies we can grow and then test for antibiotic production. So what I've done, and you're going to do this by rows this time, so 4, 4, and 3. Okay, together. You're going to take the soil sample, or in your case the clay sample. You're going to take 1 gram of it. And we're going to take 3 tubes of sterile water. These are not 99 mLs of water, these are 9 mLs of water. So, 3 tubes, 9 mLs of water. This was a question on your quiz. Okay. So, you're going to take 1 gram of soil and add it to the 9 ml tube. That's going to be a 1 to 10 dilution, or 10 to the minus
1.
10 to the minus 1. You're then going to mix that up a little, take 1 milliliter out, and make a 1 in 10 dilution of a 1 in 10 dilution. That's going to be 1 in 100, or 10 to the minus 50. You're going to take 1 ml out one more time, put it in your 9 mLs of water, and that's different types of media. So you're going to need 6 plates total. We have something here labeled GPA. That stands for glucose peptone agar. And then we have this right here, which is not labeled. This is, you should label it AIM for actinomycete isolation medium. Okay. Both those media are things that have nutrients that soil bacteria like to grow on. So this is an enrichment media for soil bacteria. We've also included 2 antifungal drugs in this media. Something called cyclohexamide and something called nystatin. We want to inhibit the growth of fungi because compared to the actinomycetes, the fungi grow very, very quickly. And we're not interested in isolating fungi using this exercise, although fungi can also produce antibiotics. We very famously know penicillium mold produces penicillin. However, that's not what we're interested in here. We already know about penicillin. We want to see soil bacteria. So we have to inhibit the growth of the fungi by adding those drugs to the medium. So only bacteria should grow on these plates. So I'm going to show you how we're going to do it. I have 2 petri dishes full of soil and 1 with the clay. So I'll use 1 spoon for the clay and 1 spoon for the soil. So you're going to take some weight paper. We have 3 balances here, so no one should have to wait too long. You're going to take the weight paper and just make a single like that. And then put it on the scale. I'm going to zero it out. I'm going to take 1 gram of soil and weigh it out. Try to avoid rocks. I think we got all the earthworms out last time. But just 1 gram, which is a lot less than you think. It's not very much soil at all. So you'll probably have to like carve off a piece of this. 1 gram. Fiddling around with it for about 20 minutes. Okay, so I'm going to take my soil and I'm going to add it to my first solution, which I've already labeled 10 to the minus 1. And you're going to get as much as possible of it in there. And then you can just throw out the meat. So this is hard to mix. And again, these are not watertight caps, so you can give it a little flip, but what I like to do is I take a pipette, because I'm going to need one anyway to do my dilution. And I'm just going to use the pipette to mix it up and down a little bit. Just like that. And then I'm going to take 1 ml out, and the dirt can clog, so just be aware of that. You might have to go up and down with it a few times. I'm going to add that 1 ml into my 10 to the minus 2. And just mix it up and down a couple of times. There. Close that up. Then I'm going to do it 1 more time for my 10 to the minus 2 to my 10 to the minus 3. Remember, you want that seated nice and firmly on there. So 1 ml out of here. I just fill it up to the 9 line. And right in there. So that's how you make your dilutions. Now let's talk about plating. You're going to plate 3 plates each. 3 glucose peptone, 3 actinomycete isolation media. But you're going to plate the same dilutions on both types of plates. The reason is sometimes they grow better on glucose peptone, sometimes they grow better on the actinomycete. We just want to make sure we get everything we can possible. So you're going to take 0.1 ml out of the 10 to the minus 1. What should I label the plate?
That's the same.
Negative.
1.
Nope. milliliter on a plate because it's too much liquid. So this is how we're going to do our plating. We're going to take 0.1 ml out of the 10 to the minus 2. That should be labeled what? Yep. 0.1 out of the 10 to the minus 3. What should we label that? Yep, 10 to the minus 4. So those are the dilutions you're going to plate. Any questions? All right. Have fun. I have everything you need. The glass spreaders are over here.
Take the plate. I can have the bottle.
Remember to label your plates EPA or AIM. Your initials,
You grab it from here?
no, I have a whole box over there. And remember, we always label the
So we're
outer
using the
side
plate,
of the
right?
plate.
That's it, right?
While you're working on that, I'm going to hand back your exam paper so you have it. If you're curious about your test score and need to think about it in percentages, just double the raw score. So a 50 becomes 100, and so on. The dilution factor, so 10 to the minus 2, 10 to the minus 3, 10 to the minus 4, your initials, and then
Just a stir. Then I gotta
these are fresh, so we're trying to
She's so big. Max.
Okay, got it.
It doesn't.
I'm sorry to interrupt you.
We hold it like.
You want to walk over here so you can have more space? Let me move this up. You
it looks pretty uniform.
Yeah, that's good. And mix it. But why you use two plates?
Because we have to use our heat.
Oh yeah, AC.
when you're done, just remove the labels off your solutions with alcohol and put them in the discard. And then you can clean up, take pictures of your Winograd columns, and have a fantastic weekend. I know it's been a super busy week. Enjoy whatever break you can get for yourself. And I'll be in here for a few minutes after class today, finishing my setup if you have questions. Can I keep my plates with you? Sure. Thank you. It's one of the hazards of being in the front row. What's up? If you find your plates have a lot of liquid on them, what I'm going to say is store them in your drawer agar side down until next week when you can flip them over. Just to allow the liquid to soak. If you already flipped them and it's fine, it's okay. Glass spreader, just like you did for the viable plate count. You can just put it back in the spreader. Yeah, if you're done with it, you're done. Good call. We don't want to start with this or anything. Okay.
No, these are can.
Yeah, just take the labels off with some alcohol. And then, yeah, you're done. Make sure you put your carbohydrate fermentation tests in your drawer, lay down your bench. Have a wonderful weekend.
And can you got photos of the?
Yes, I got home. She's got it.
I took time from you.